missing. Okay. Then we go over to the next talk by Wolfgang Wenzel from Karlsruhe, modeling bio-nanomedicine interaction.
Thank you very much. Now, I think this is amazing. I think both of us have been to this conference a couple of times, and to my knowledge, this is the first time that we have more than one theory speaker. Right? So actually, we have a theory session now. Yes. Which is quite a development. So can I ask into the audience, how many of you?
I insisted on it.
Huh?
I insisted on it.
Yes.
To have this session.
Yeah. so how many of you are theorists in the audience, and how many? Okay. There's maybe some of the speakers already left here. Okay. So since I have now more time, I will just keep on talking forever until Gianco stops me, and I will tell you a little bit about modeling bio-nano interactions. I will not focus on one particular study because I guess we have time in the coffee break afterwards to discuss the details. What we have developed basically over the past decade is a very efficient method to formulate, to simulate the structure formation and equilibrium conformations and binding properties of small peptides. So this is the workhorse of all the things that I will talk about today. And what you've just seen here is kind of a folding process of a small peptide of about 30, 40 amino acids. The backdraw of this method is that it uses an implicit solvent model. That means that you do not really represent the environment in an explicit way, which means that we are pretty much limited to room temperature, pH 7, the things we know and love. Even though we are now tending to improve this a little bit. Now, okay, using this technique, we have done quite a lot of work on protein modeling, drug design, nanomaterials development, and a large number of applications, and some of them I would like to share with you today. The first spin-off of this methodology is, of course, a docking program, which we call FlexScreen. It pretty much goes in the same direction as we've heard in the first talk, and it enables you to very rapidly screen large libraries of small molecule drugs against structurally known protein receptors. We can incorporate the flexibility of the drug, but also the flexibility, at least to a certain degree, of the receptor. And this can be applied not just to small molecule drugs, but also to a limited extent to peptides and other nanoscale objects such as nanoparticles, vesicles, and the like. So to give you an idea, we started with a study together with a group in Spain to look at anticoagulants. So here is the overlay of the docked and the X-ray structure of heparin to its receptor. This was then run against the zinc library, which is a publicly open database, which has about 8 million compounds. This takes a good 2 or 3 weeks to run, but after that we were able to identify 20 high-scoring molecules. Since we don't really have the synthetic capabilities which larger institutions have, in particular the industry, we could only take those that we could buy, and out of the 6 basically that we could buy, we were lucky to identify one nanomolar inhibitor, which was active in vitro in mice and in the blood of heparin deficient patients. So this kind of gives you an idea what sort of in silico screening in the best of cases can do. So the next step, what we wanted to look at was basically hERG channel blockage. Many of you know that the hERG channel is one of the primary toxicity measures for drug approval processes. And we first started to validate the model. That is, we generated a homology model because there's no crystal structure for the system. And then we basically docked a bunch of small molecule inhibitor with various affinities. We were able to basically rank the affinities in the correct order. And what is more importantly, For some of them, there are quite extensive mutation studies, which even in the absence of the structure tell us which are the key residues where the ligand and the receptor interacting, and these could be also validated. So once we had this, we felt a little bit brave, to go on towards larger objects, and what we wanted to investigate was the nanotoxicity of small gold nanoparticles. These are basically clusters of a size of just 1.4 nanometers. They comprise 55 gold atoms. They're not stable in solution. So, what sounded like a drawback at the beginning is that they have to be stabilized by certain ligands, and the chemists have a sort of choice of to what ligands they want to use. So there's typically some chemical bond, in this case phosphate, could also be sulfate, to the gold nanoparticle. The experimentalists performed some screening where they basically saw that the hERG channel, and that is kind of interesting from a regulatory standpoint, was blocked by the gold nanoparticles capped with a ligand shell of TTP-MS, while they were not blocked by a ligand shell with with gold nanoparticles blocked by TTP-PS. So these just differ, essentially by small chemical differences of the ligand. And because they didn't understand that, we started to do simulations. We could indeed dock the nanoparticle into the receptor, we could dock it, but we had to use actually brute force, right? These docking processes usually cheat a little bit by guiding the ligand towards the receptor pocket, because otherwise in finite time you would never bind. And we found that we had to increase this guiding potential for this particular case to such an insane extent that we started to worry that the physical model was correct, right? And indeed the physical model is not correct, right? The channel absolutely hates this nanoparticle, right? And the only reason that would explain why the gold nanoparticle would bind to the channel is ligand displacement, right? And then that explains also the experimental finding, because the two ligands that I've discussed in the experiment differ in their affinity to the nanoparticle, and therefore only those nanoparticles bind, and then they bind irreversibly, which are able to displace the ligand among the binding process, right? Okay. Next, once we had discovered this, we thought, well, is the same effect also present in the study of the interaction of nanoparticles with cell membranes, right? Because often, and I think this is discussed here in this conference quite a lot, we know that the properties of the nanoparticle interacting with the biological system are not primarily affected only by the physical constituent of the nanoparticle, i.e. whether it's gold, silver, platinum, or whatnot, right? But also by the environment, and often the dynamic environment the nanoparticle forms in its biological system, right? So we synthesized a bunch of nanoparticles ranging from 1.4 nanometers to about 50 nanometers and decorated them with a bunch of cells, with a bunch of ligands, and then exposed them to artificial lipid membranes in what is called the black lipid membrane experiment. That is what you've seen here, and it is my only excursion as a theorist into the world of experiment, and after doing this for 4 years, I promise you that I won't do it again. What you have is you have this double chamber, and then you have a pore, and in this pore you place a lipid bilayer, and the experiment derives its name from the fact that this bilayer is black when the membrane is intact. Then you put in the nanoparticles on one side, and you apply a current from one side to the other, and by analyzing voltage, and by analyzing the current, you can basically analyze the permeation of the nanoparticle, right? And so what we found is the same thing that we had seen for the nanoparticles interacting with the hERG channel. The TTPS nanoparticles started to permeate the lipid membrane at a radius which is larger than 4 angstroms, meaning that the 1.4 and 4 angstroms didn't go through, but 5, 10, 12, 15 went through. while TTP-PMS started to permeate only at basically twice the radius, right? And this can again be explained by an analysis of the affinities of the ligand shell to the nanoparticle. For TTP as coded nanoparticles, it is much easier to strip the ligand of the nanoparticle, therefore they incorporate into the membrane much easier. The naked coated nanoparticle is much more hydrophobic than the ligand-coated nanoparticles. That means it will stay there once it is there. We also don't see basically the nanoparticles come out on the other side of the experiment, while the nanoparticles coated by the other ligand are much more, while the other ligands are much more strongly bound to the nanoparticles, so they basically don't come off. The nanoparticles do not incorporate easily into the membrane, and therefore you see no current. In another study, we look now at the interaction of peptides with membranes, using always basically the same simulation methodology that I've shown you before. We extended the model for the physiological aqueous environment towards an implicit membrane model, and we studied protein translocation. As many of you know, there is two translocation mechanisms that are prevalent in cells. One of them is the SEG pathway, where you basically have the pore inside of the cell membrane that is roughly the width of an unfolded amino acid chain, right? And then, in order to get protein translocation, you tag the protein with a recognition sequence. It is transported to the cell membrane. It is unfolded there and then exported to the extracellular environment. This doesn't work for proteins that bind cofactors or that need chaperones to fold, right? And so therefore, nature has figured out another less widely used way to do this, which is the called TAT-mediated translocation process. And in this TAT-mediated translocation process, you actually make a pore in the membrane, which is the size of the fully folded protein. Then you export the protein through this pore, and so the fully functional protein, including cofactors and other elements, is exported. Right now, of course, the cell would immediately die if you had a permanent hole of that size in the membrane. So there must be some dynamic measure to constitute the pore when it's needed to export the protein. And this is done by the so-called TAT-ABC complex. TAT-C is a huge regulatory protein for which we have a crystal structure now since 2014. But what TAT-A and B are small peptides, but what they do was basically not known, and it's difficult to analyze because it's a transient process. There's no crystal structure and the like. Right? So what we did know, and what the experimentalists had figured out with solid-state NMR, is that if you look at the peptide, then it comprises a large hydrophobic helix, which embeds into the membrane, as you would expect, and then a large amphiphilic part, which nobody really knows what it's good for. But when you analyze basically the residues of the amphiphilic part, then you realize that there are patterns of salt bridge acceptors and donors that you could piece together to make salt bridges either intra or extra molecular, right? And so this is, for instance, one pattern how you can connect the salt bridges on one of the peptides to another one, right? And there's literally thousands of these patterns, and this is what the biologists sort of had come up with, that connecting these salt bridges might lead to the formation of intermolecular contacts that may then lead to the formation of the pore. Right? So what we could do is basically make a model of these peptides, right? And in this case, we assumed that the hydrophobic helix would stay hydrophobic. The rest of it we kept pretty much free, right? And then we did simulations where we could, in the membrane, assemble complexes of different sizes that would try to match the constraints that we basically dreamt up on the basis of these salt bridges. Most of them just fell apart, right, simply because the salt bridges are not compatible with any structure in the membrane, right? But there were a few cases where we could actually assemble a complex. These complexes then matched with what is known from cryo-EM. Sorry, there's a slide missing. Then, some very brave PhD students went on for 3 years, and they generated point mutations in these membranes to, either switch off some of these salt bridges, in which case the complex would fall apart, or to switch them back on, right? Even generating salt bridges in places where there hadn't been salt bridges before, and that enabled us to essentially validate the model, right? So now we have the model for the pore. We have a crystal structure for the TAT-C protein, which kind of generates or guides this process. But what we don't know at the moment is how these two hang together. And finally, I want to go on, and discuss a little bit the functionalization of surfaces of nanoparticles with peptides, which is relevant for biological applications, but also in biotechnology, because increasingly we would like to know which peptides bind to certain nanoparticles, and whether we can design peptides to bind to nanoparticles, right? So the bad news, if you want to try to do this, is that there's essentially no experimental data for the affinity of individual amino acids to all kinds of surfaces that we're interested in, such as metallic surfaces, such as silica, such as iron oxide magnetic nanoparticles, and so on and so forth. And the reason is that there is no measurement techniques that have the resolution to detect individual amino acids binding, right? And if you know of a method, please let us know. We spend a lot of money and time to try to get this under control, right? So one of the ways to do it is to deal with molecular dynamics simulations, a little bit of the type which we've seen in the first talk. But as you can see here, these give very different results for the same amino acids and the same surface, right? So it is essentially at the moment the input data which is problematic, right? But once you have the input data for the individual amino acids for this implicit model that I have discussed, which again here we have extended to incorporate the surface effects, then you can, in a good day, quite easily compute for peptides, the free energy of binding to the given surface. You can check the surface conformation in the implicit model versus that of the explicit model, which was done previously, and then go off and screen a large set of peptides, including conformational flexibility, with reasonable amounts of time. So one of these simulations for peptide of length 10 takes about 3 or 4 hours, right? And with that, at least we have now a protocol that distinguishes the gold binding from the silver binding peptides. The correlations between experiment and modeling are not super good, but also the experimental data is not super good. And we have implemented an iterative model to improve, we are able now, in the availability of experimental data for peptides, to improve the model iteratively. So with that, I hope to have given you some sort of overview of what simulations can do in the study of bio-nano interactions. I would like to thank many collaborators and all of my students who did this work, and I thank you for your attention.
I think it's open for discussions, questions, remarks.
Hello, nice talk. I was wondering, when you do your simulations of absorption of amino acids on surface, what would be the effect of not considering an infinite surface, but a nanoparticle as per se?
Well, it depends pretty much on the size of the nanoparticle, right? I mean, even a 10 nanometer size nanoparticle is an almost infinite surface for the peptide, right? So if, unless you're talking about like an antibody or some humongous protein then, essentially, assuming a flat surface makes no difference.
Mm-hmm. Yeah, because we have simulations and we see a lot of effects, of course, for the small ones.
Yeah, for the small ones, that's different. And of course, at that point, I mean, if you take the gold nano, these 1.4 nanometer particles, right, then they have of the order of, let's say, 50 small molecule ligands on top of them, right? So one of these ligands is comparable to an amino acid, right? So it's very clear that, a peptide of length 10 would, differentiate, basically the size of the nanoparticle.
Okay.
Yeah.
And does the model include like the effect of sortation layer? Because it's implicit solvent, right?
Yes, and so the idea of the implicit solvent model is the following. You have to get some sort of data, either from experiment or from an explicit solvent simulation for the individual amino acids, right? And so for that, you have these delta G curves, and then you train essentially the implicit model to reproduce that. Right?
Okay.
Okay, and then you use that going on, right? And if you don't have enough data, you can refine this by throwing in additional data for peptides later.
Okay.
Okay.
Thank you.
You're welcome.
Thank you for the nice talk. I just wanted to see if you try and account for the effect of glycans and other structures which are found on proteins but are not part of the primary structure of the protein in your simulations.
On the peptide or protein side, we're pretty much flexible because the protein is always represented with atomic resolution, right? So if you tell me basically where your protein is glycosylated, that would be possible to incorporate in the simulation. But then again, to put one more caveat, you would have to also give me some interaction profile, right, of the sugar with the surface.
More questions? Yeah.
Thank you for your talk, very interesting. And you mentioned you use a flex screen for docking.
Yes.
But this was, I did not pick up if it was organic molecule peptide docking or a peptide protein. I did not understand. It was in the beginning.
Basically just for small molecules. It's an optimized module in the, Simona package. Our package is called Simona, right? And there's a module called flex screen, which does the small molecule peptide docking. And it uses some tricks, right, which you want to use for docking of more or less rigid objects. Right. So, but the basically concept of how the simulation is done, namely Monte Carlo versus molecular dynamics, and that we always use some sort of implicit solvent model. That's the same for all the things that I've talked about today.
Thank you.
Yes. If no more questions, thank you very much.